So let me do the screen share. So we're going to be doing the this bioassessment lab today. Real quick, I've graded all the past lab assignments you guys had with me. I think most of your grades are in. It's not that close to finals, but you do want to check those grades because if you have any questions or want to refute anything, you know, the sooner you do it, the better. So just make sure that that's all correct, and let me know if you have any questions. You guys should be seeing my computer screen, I think. And feel free to stop me if you have any questions. We should get through it fairly quickly today. Hang on, Levi, I'm gonna make you co-host. And yeah, just if anyone pops up, you'll have to admit them for me. So as I said, we're going to be talking about bioassessment today. So bioassessment is simply an evaluation of the biological conditions of a water body. Generally uses a survey of the resident biota. And these resident biota can be thought of as a continual natural monitor of environmental quality. And they'll respond to both short-term and long-term trends in pollution or habitat alteration. So by assessing the survival, reproduction, and abundance of these biota, we can get indication as to whether the stream is impaired or not. So if you remember back to, I was talking about feeding guilds in the previous lab I taught. So if there's a particular macroinvertebrate or whatever that occupies a feeding guild, if that feeding guild's not represented, that might be impaired. If there's a species that should be there that's not, it might be impaired. And the goal is simply to preserve the biotic integrity or the capacity of the watershed to maintain a balanced community that reflects the natural assemblage of organisms and functional organization in that region. So these generally incorporate numerous variables. It might be, you might survey macroinvertebrates, fish, microbes. You might be looking at richness, abundance, or kind of the overall biomass. You might look at the reproductive success of various organisms, or you might be looking at toxicology. So is there something in the river or in the organisms that's toxic, and what's the concentration of that toxin? And there's many more variables that you might cover depending on the bioassessment. So we talked a lot, I think, in the aquatic organisms lab about aquatic macroinverts. So this is just the way you may sample those. So you would have what we call a kick net, usually has a flat bottom, and you put it on the bottom of the stream, and then you stand upstream of it and kind of just kick the gravel and substrate around, usually within a 1 square meter area. And that'll free up all the macroinverts that are in the substrate. They'll drift downstream and get caught in your net. You can then take them and dump them out, sometimes into something like this, and you'll just kind of pick grids randomly until you've sampled to get 200 individuals. So you'll just be randomly sampling these grids and identifying the species or orders. Actually, generally, I think they do it to the family level because it gets pretty difficult to go on species level in each of those. So sorry, my monitor is blinking out. So bioassessments will include different types of metrics. So some community metrics might be the loss of sensitive taxa. Common metric for this is like EPT richness or percent EPT. So you remember in the aquatic organisms lab, we talked about Ephemeroptera, Plycoptera, and Trichoptera. Those are three orders of macroinvertebrate species that are sensitive to pollutants. And so what you'll be doing for the EPT richness is you'll be counting the species within each of those three orders. And then for the percent EPT, it'll be the percent of organisms within those orders versus the entire sample. So all other organisms in the sample. And on the flip side of that, you have the increase in tolerant taxa. So it might be the dominant, how much of the sample is made up of dominant taxa or tolerant taxa. Generally, we look at the top two dominant taxa. So multi-metric indices of biologic or biotic integrity. These are sometimes referred to as stream condition indexes. We'll be talking about the West Virginia Stream Condition Index down here, abbreviated WVSCI. So it can be a combination of metrics. It relies on first characterizing the reference conditions in a region that these are generally as close to pristine as you can get. And you're gonna identify the key biological attributes that are changing in the stream that you're concerned about. And then you'll choose metrics based on like either species diversity, the functional feeding group representation, or the abundance of pollutant tolerant or intolerant species that will respond to changes in those. So basically, we're looking for a region-specific group of metrics that we can use to identify if a stream is stressed or not. So the West Virginia Stream Condition Index, they did a study here to determine which streams are a way to identify streams that are stressed by human disturbance or pollution, and they came up with these 6 metrics. And these over here in brackets is how these metrics will respond if the stream is disturbed. And obviously there's magnitudes of that response, right? So total richness, just the total number of species present. EPT richness, I already covered. So that's the number of species within the orders Ephemeroptera, Plycoptera, Trichoptera. So your mayflies, stoneflies, and caddisflies. And then the proportion of those organisms that are EPT in your entire sample. The fourth one is percent Chromonidae. These are your non-biting midges or lake flies. They're pretty tolerant generally. Then the percentage of the sample is made up of the top 2 dominant taxa. And then this Hilsenhoff Biotic Index is the 6th one. So this Hilsenhoff Biotic Index weights each taxon in a sample by its proportion. So its abundance and its tolerance value. So each species is given a value on a scale of 0 to 10, 0 being intolerant and 10 being tolerant. And it just comes up with a value for that based on the abundance and its relative tolerance. And And then each of these 6 metrics are standardized on a scale of 0 to 100. And then they're averaged. So we'll have one score, which is our West Virginia Stream Condition Index for each stream. So we're averaging all 6 of those. And then if it's less than 65, it's considered impaired. If it's greater than 65, it's not impaired. This is how our state defines whether or not a stream is impaired. So that's just kind of general overview of the Stream Condition Index. So we'll be using data from the Cheat River watershed. It was collected by West Virginia University graduate students. And these streams are impacted by acid mine drainage. So we'll just be, I gave you an Excel file. We'll be going through that. It'll calculate all those values for you that I just went through. And we'll just be making some figures, and then there's a few discussion questions for you guys at the end. But we'll be working through most of that today in class. So just a real brief overview of acid mine drainage. This is kind of a common problem here in West Virginia because we have a lot of old coal mines, and water comes out of those and seeps into the streams. And it has a high proportion of iron in it. Iron dissociates in water and forms sulfuric acid. So it's like, I don't know the chemical or the compound name, like iron sulfate, maybe, or iron sulfide, probably something like that. But this is producing basically sulfuric acid. So this decreases the pH in the stream. If pH increases, a lot of times what they'll do to mitigate the low pH is dump calcium carbonate or like just limestone into the river, like just a huge dump truck and dump it in. And that'll make it more basic, but then these heavy metals precipitate out. And then it also increases the specific conductivity of the stream as well. So are there any questions on what I just covered? No. Okay. So next thing, you should have this spreadsheet. It's bioassessment spreadsheet, students. So this first tab, this site data, is where we'll be putting all of our data or all these values that we're going to calculate. The abundances tab is our raw data. So you can see we have our taxon here. Across the top, we have our site name or a stream name and stream code. Really pay attention to the stream code because you'll notice there's a few. Muddy Creek was sampled twice, but the stream code is different. So you're going to want to make sure that you're getting these in the right place. So I pay attention to this stream code that's here in red in site data. It's called AND code. And we did put those in bold, those ones that are tricky, but there's a few other names that are kind of similar, and they don't go in the same order here as they are in here. So what we're going to do is we're going to just start with column B. We're going to take the first one, Beaver Creek. You can just take the whole column, Control C, then go to this WVSCI calculation tab. You notice here we have the taxon, so this is all the same. You're just going to select this column B and then hit Control V. You'll notice it updated all of these values over here. Hopefully this is working for you. So I took these formulas here, interposed them into this. So we can just highlight these cells here, Control C. And I want to make sure we were using Beaver Creek, which is 12B 1.1. So I'm going to go site data Beaver Creek 12B 1.1. I'm going to highlight these cells. Then I'm going to right click and paste only the values. If I were to control V, it's not going to work. It's going to paste the formulas and it's not going to understand. So I need to do the values. And I'm just going to go through and do that for each of these. And I can just highlight Control V over top of it, and it'll update automatically. And make sure you're getting the correct one. So if you guys want to start going through this and doing that, then we can move on afterwards. So Daughtry Run 19, it's way down here. So I'm going to let you guys go ahead and start on that. And let me know if you have any questions. And what I'll do, so I'm going to create a breakout room. And as you get done, I'm going to let you go over to that breakout room. And Levi will take you through creating the figures. And then once everyone's done entering the data, we'll circle back and group up. So you should be able to choose if you want to go to the breakout room. Oh, hang on, I have to open it. Otherwise, I'll be here to help you if you need help with the data. And we should get through most of the homework, if not all of it today. I think we got done a little early in the last class too, so. Yeah, and please don't leave until you at least get a start on the figures. Once you've figured out the figures, if you don't want to stick around to make the rest, or you don't want to discuss the discussion questions, then you can leave. But make sure you at least get a start on those figures.
So once we get all of the data in, or what do we do then?
You can go over to room 1, and Levi will help you with the figures.
All right, cool. Okay, I do.
Yep.
I just finished up all the. I kind of mixed it up at first, so it took me a little bit to change it back. But it's not giving me an option to go to the breakout room. So I don't know if you could put me in there.
Yeah.
Thank you.
Yep. No problem. You working through everything okay, Miranda?
Sorry, I did the first data transfer the right way. I had to ask somebody for help because I missed you doing it because I was trying to pull it up.
Okay.
But I don't know what I'm doing wrong. When I try to do a new one on top of to replace the old one, it's just not going through all the way.
Interesting. Okay.
When I try to paste the second abundance column into the calculation tab, it only goes halfway down, so it doesn't change all of the numbers.
I see. So you might be scrolled down. Are you hitting just the whole column? If you select B at the top and then paste it Control V, does that work better for you?
Let's see.
Otherwise, I can have you share your screen.
I'm actually doing it on a separate computer sitting
Oh,
on my
are you?
desk. So.
Okay.
Yeah, it's still not going. It still only takes it halfway down for some reason.
Interesting. So you're not doing anything like this. You're selecting the whole thing. So see, I was gonna do directory run, Control C. It has that whole column. Then B, Control V. And what's it doing? It's only going down part of the way.
Yeah. So it's only going to row 31, and it's putting the sum there instead of all the way down.
Interesting. So something weird must have happened.
Oh wait, I got it.
Okay, good.
Cool.
What was the issue?
I wasn't hitting one of the boxes.
Okay.
I'm just not very good at this.
Neither am I. Excel's tricky for some reason. They tried to make it too easy. Now it's hard. As long as it's working. How many of these did you get?
Only 2 so far because I restarted it twice trying to get it to work.
I gotcha.
All right, I got all of them done.
All right, cool. I'm gonna close the rooms. So I'll go ahead and I'll just give you the demo on how to make them. And
Okay.
that way it'll be in the recording. So what you're gonna do is you have the, hang on, where is it? Oh, here it is. So the homework assignment, what we just went through was number 1. So for 2 and 3, you're going to be making different figures. I'll work through 2 and it'll be basically the same. It tells you which is the X and which is the Y. So the
Hey,
first,
Alex, sorry
yes.
to our breakout room just like
I ended that so that we could all come back together since everyone's done entering data. And I figured if you're still working on the figures and have no questions, you know, keep going. I'm going to explain it to Miranda so that it's on the video.
Okay.
And yeah, so just select a blank cell anywhere over here, go to insert, and we want a scatter plot. Then just select this and it should open it up, but if not, go to chart design, select data. So the first figure 2A, we want conductivity on the Y and pH on the X. So I'm going to hit add. I'm going to select this little arrow here where it says X value. And I'm going to highlight the pH column. I'm not going to include Dunkard Creek because it has NAs in it. It's our unknown. I'm just going to hit enter. And then for the Y values, I want conductivity. So again, selecting that column, excluding Dunkard Creek, hit an OK, and then OK again. I won't need this chart title. I need axis titles. So I have pH down here on the X and conductivity on the Y. And you don't have to do this. I don't like those, so I'm just going to click them and delete them. The next thing we need is a trend line. So you hit this little plus and go to this arrow here, trend line, and more options. If you don't have this, like you're on Mac or whatever, I think you can go up to add chart element, trend line, more trend line options. So this should pop up, format trend line. You want to click on these bars. You need to display the equation and the R squared value. You'll have to move it up to where you can see it. And then once I've done that, I'm no longer on the line, so I need to click the line again. You'll see these 2 little dots at either end that'll show that it is selected. And now I want to pick the best line. So I want to try to get this R squared as close to 1 as possible. And the options it tells us to look through are linear. R squared is 0.5, exponential, 0.3, log, 0.6, so that's the best so far, and power, 0.4. So I'm going to choose the logarithmic version. So that is the first graph. So that is 2A. Now, if you want to kind of go quicker through these, you can select this figure, Control C, click a new blank cell, Control V. And now I have that same figure. If I select it again, sorry, my monitor's blinking out. Go to chart design, select data, it brings up this thing again. I'm going to select series 1 and hit edit. Now, the only difference between 2A and 2B is instead of conductivity on the Y, I want aluminum on the Y. So I'm going to do this Y series value, hit this, and then highlight these cells, excluding Dunkard Creek, hit enter, and hit OK, and then OK again. And then I have to change this to aluminum. And then I can select my trend line here. It automatically comes up. Now I can pick the best one. So I'm on logarithmic. And the R is 0.72. I looked at power, it's 9.2. I imagine I'm not going to get better than that. And down, so I'm going to just choose a power. So you can do that. And then, so it's the same thing for these next graphs, except what we're changing is the y-axis to the WV SCI. So that's this column here. So hopefully that makes sense. If you get that, if you feel comfortable, you're more than free to leave. If you want to discuss the discussion questions, you're more than free to ask questions about those. If you want to work on it, you know, and finish it up, we'll be here for that too.
For question 3 and the WVSCI stuff, do we include Dunkard Creek in that as well?
I don't think you know, because you won't be able to use the pH, like any of those earlier variables. They're all NAs. I don't think it'll work. So don't.
Alrighty, thank you. Just making sure.
Yep.
And I also say that when you go about plotting your different sort of environmental covariates on the x-axis with the percentage EPT, due to having 0% EPT for some, you're going to take away your ability to plot using certain, or add certain trend lines that basically it would be impossible for you to get a value of 0 on the y-axis.
Yep.
So don't include those?
No, you include those. It just takes away. So like on the problem set, it asks you to fit a trend line using either a power, linear, logarithmic, or exponential function. But you know, when you use exponential and power, they're essentially using, you know, exponentiated data. But when you think about it, you know, anything to the 0 power, so 1 to the 0 equals 1, 2 to the 0 equals 1. So there's no way. And then if you get into a negative exponent, it just essentially approaches 0, but can never reach 0 on the y-axis. So you'll still be able to choose between, say, a linear and a logarithmic. But I'm just saying that eventually you might come to a point where you're trying to decide between trend lines and it's not going to give you the options for some. You're not doing anything wrong. It's just because there's zeros for the percentage EPT.
Okay, thank you.